and welcome to day 28 of MetPhys 2020. I'm coming to you live from Montreal with Jennifer Estall. All right. So, as I've said on multiple occasions, metabolic physiology in isolation represents a new way to run conferences in an inclusive global manner. We're able to reach out to anyone, anywhere across the globe, and it's cost effective. The White McGill Lab is happy to take charge of all the costs related to this conference, so anyone can attend at any time. It's inclusive, no matter where you are, what your scenario is. You can listen to these talks at your leisure, and it's green because we're not getting on flights to go and end up in places. Today is the second to last day of the series, but don't worry, there are a lot of other series that are just launching or have been going for a little while, but will go out until August. A lot of these series have 1 talk per week or 2 talks per week. I've talked about their links yesterday. Most of the ones that I'm showing right now are actually listed in YouTube. So if you go to YouTube, you can subscribe to them. You can see that I've subscribed to a collection already: Global Immunotalks, Cardiometabolism seminar series, Lance Johnson's Alzheimer's Afternoons, Kelly Macklis's Blood and Bone, the Immunomet, and the STEM Village. And when you do that, if you click on the subscriptions link in YouTube, you'll get a collection of all the talks that are happening in the past week, the past month, and all the ones that are on today. So if you subscribe to us, you'll also see ours in there, and you can watch those at any time. But it's a nice way to kind of catch up on what's available in your science. Cassidy, now is your time to take your image. This will be the last one for Inception Challenge. Tomorrow I'm going to show every single one of your images. Her idea yesterday, she said, for a seminar series was one about cats. I completely agree with that. And I think that's a neat way to go. We launched a vote yesterday for the best mug, MetPhys 2020 mug off. It's very, very close. There's about 30 minutes left. So get in and make your vote on Twitter. It looks like, last time I looked at it, Macrophages of T-Rex had actually taken the lead. It looks like Thierry Akier's strategy of only giving 1 cup to 1 person might not be making him any friends in this vote, but we'll see if he can hold on to win, and I'll let you know tomorrow. If you want to ask questions during the series or after the series is over, you want to reach out to the speakers, the nice way to do that is using the hashtag MetPhys2020 on Twitter. And then also adding the speaker's handle. You can see that Carolyn Cummins' handle is her name. Today's handle is DNA Chicken. Hopefully the speaker will enlighten us on what that actually refers to. You can learn those, you can find those, and you can link them. If you don't know the speaker's handle, just use MetPhys2020, and I'll add them so that they know what's going on, and they'll be able to get back to you, or maybe the postdoc in the lab that did the work, like yesterday, was able to answer a lot of questions. I thought that was really great. None of this series would have been possible without the wonderful speakers who agreed very rapidly to take part, but also who are very open with their science. I know a lot of people think, well, if this is going to be online, it's going to be listed. No one's going to share anything new or of interest. It's all going to be published work, and everyone has kind of rebuked that old model, and I think that's what's made MetPhys 2020 very exciting. We've got state-of-the-art talks, as you would a Keystone or Gordon conference, and available to everyone. And so that's why we're going to renew this next year for MetPhys 2021. And so tomorrow will be our last day. Yes, we're staying in Canada, but heading to the other coast to UBC, where we'll hear from Ramon. If you want to thank me for this series, the way that would really honor me is if you looked up the Pathway to Stop Diabetes program. And 1 second, my battery is running low. I'm back. There we go, move over, Jack. Thank you. It's the only place for two of us on this board. You can drown. If you'd like to thank me, you can look up the Pathway to Stop Diabetes program. They support my lab and they support a lot of other great work. Salim Riera, who spoke a week ago, gave a fantastic talk and wouldn't be here without the generous support of the ADA. So if you have the means to do so, that would be really an honor. All right, so tomorrow our last talk will be from Ramon Klein-Geltneck at UBC, speaking about glucose metabolism in CD8 T cells, conditioning a tumor-killing army. I think that's going to be an amazing talk and a great one to finish off our sessions. I've really enjoyed all the interaction online and Twitter, and I'm looking forward to catching you all in the Twitter space after this talk and during the talk. All right, so without further ado, I'm going to kick over to Jennifer Estall, and I am about to make you the host. Making you the host right now. I'm going to stop my share and stop my video. All right, we can see your screen. We can see you.
And you can hear me?
We can hear you. All right, see you in the Twitter space. Thanks, Jennifer.
Thank you so much, Phil. It's great. Thanks for the invitation to be part of this. It's really been amazing to watch the talks and just be a part of and asked to be a part of this series. Now you've put me under pressure to explain my Twitter handle at DNA Chicken. I wish it was a more interesting story actually, but when I was in undergrad, I lived in a residence at university, and we were all getting to know each other at the beginning. And my family, on my mom's side, is originally from Quebec, and so they wanted to know what her maiden name was, what the family name was, and I said, "Oh, it's Poulain," and they said, "It's chicken," and I said, "No, it's Poulain." But as many of you know, Poulain sounds very much like poulet. And so for some reason since then, I was known as Jenny Chicken my whole undergrad while I lived in residence, and it just kind of stuck and it got written on my door. And so it's kind of a nickname that my close friends have always called me from school. And so I used it as my Twitter handle, not realizing that Twitter would become as popular as it was, but I won't change it now because it's how I'm known. Today, I'm going to talk about the work that we're doing in the lab, of course, in terms of transcriptional regulation of metabolism, and how we kind of stumbled on how a pathway that we've been studying for quite some time now touches on inflammation as well, and how this might be involved in the pathogenesis of fatty liver disease and diabetes. Oh, and now I can't advance my screen. Okay. Okay, I'm gonna have to do that. Okay. So our lab has been around for about 10 years now. We're really interested in the pathogenesis of diabetes, and we're not particularly involved in studying the endpoints of the disease, which is really beta cell failure and the lack of insulin production and secretion, but we're more interested really in the 49% of people who are walking around right now who don't know they yet have diabetes, and the changes that are happening in their body at this time that are eventually going to lead to the disease, which involves a dysregulation of glucose and insulin resistance. We know now from many studies in the work of a lot of people that diabetes is a disease of multiple organs. It's not just the pancreas or the pancreatic beta cell, but it also involves really an intimate relationship between muscle, liver, adipose tissue, the pancreas, the brain, the intestine now. So our lab particularly focuses on the liver and potentially the role of non-alcoholic fatty liver disease in the pathogenesis of diabetes, how this is involved in not only glucose regulation, but dyslipidemia, how the liver can talk to the beta cell, how it normally talks to the beta cell, how that goes wrong in diabetes. And while we are really interested in the early stages of the disease, we actually are interested in finding out how this kind of altered metabolic environment that seems to then lead up to diabetes also seems to predispose a lot of people to the development of certain types of cancer. So how cancer pathogenesis is also related to the pathogenesis of diabetes in terms of common molecular mechanisms. So this is kind of the system we focus on mostly in the lab. It's really the interconnection and intercrosstalk between the pancreas and the liver. And these two organs are really in sync with each other. In a normal, healthy human being, these two organs are really the main glucose sensing organs in the body, which means that they are constantly monitoring how much glucose is in the blood and regulating that glucose so that it stays within a very tight range of 4 to 5 millimolar. This is a very healthy range. When you get too high, it starts to become toxic for tissues, and that's eventually what leads to diabetes and other metabolic diseases. So when we eat and the glucose in our bloodstream goes up, the pancreas, of course, responds to that glucose by releasing insulin. This insulin is then immediately transferred to the liver through the portal circulation. The liver is one of the first organs to see insulin and to respond to it. The liver actually is responsible for clearing half of the insulin. So half of the insulin that the pancreas secretes in response to glucose is actually eliminated by the liver before entering the rest of the body. So it responds in terms of insulin signaling, but also in controlling insulin concentration. The pancreas also secretes glucagon when we're fasting, and that's really the main signal to the liver to transform, to go into a more catabolic or energy producing state. And in addition to glucose, the liver is also being exposed to the lipids that we are ingesting, amino acids that we're ingesting, and in addition to nutrients, the liver is also seeing all of the metabolites that we are also absorbing from our gut, and those are nutritional, but also other metabolites that are secreted or produced by the gut microbiota. Byproducts of the gut microbiota, such as lipopolysaccharide, can enter the bloodstream as well as bacteria itself. So the liver, in addition to being one of the main glucose sensing and nutrient controlling organs of the body, is also responsible for really surveying the blood that is entering our system, looking for toxins, eliminating toxins, responding to external stimuli, responding to what we're eating. So it's a really important kind of first pass organ to help us maintain proper metabolism and energy in our body. And this whole system, which is really tightly linked in our body and really rapidly responding, goes wrong in diabetes. We know that there's multiple things that can happen. One of them in particular is that the liver starts to produce inappropriate levels of glucose when it shouldn't be. And that can lead to then, or contribute to the hyperglycemia that you see in diabetes. The liver stops responding to insulin in the appropriate ways. In ways it becomes insulin resistant in some ways. In other ways, it starts to accumulate lipid or produce too much lipid. It doesn't deal with the lipid and the amino acids and the glucose appropriately that it's seeing from our digestive system. All of these can lead to the development of fatty liver disease, which is an asymptomatic symptom where you start to see buildup of fat inside the liver. And so what we're trying to understand is, how does this affect the metabolism of the liver cell? How does it affect the metabolism of glucose and the rest of the body? And really, what is the importance of the hepatocyte in this whole system and how it talks to the rest of the body? We all know that the molecular mechanism, so while we do a lot of physiological studies in the lab, we're also doing a lot of cell biology. I'm really interested in molecular mechanisms. And we know that in diabetes, it's really multifactorial and complex. So we all kind of have our favorite pathways or favorite mechanisms to look at. We can be looking at, insulin resistance, mitochondrial disease, inflammation, microbiome, lipid metabolism. In our lab, we focused for many years on mitochondrial dysfunction and whether or not it played a role in the pathogenesis of diabetes, so really early on in the disease. Because it was known from many early studies in humans, so this is human liver here, that mitochondria will become swelled, they'll become dysfunctional. You can see that the structure is not there, the cristae, you can hardly make them out. We know that the function of mitochondria is not appropriate, but these are often tissues that are taken in human beings or in animal models really late in the disease, where the insulin resistance is already developed, the glucose intolerance is already developed. So we're not sure exactly whether this is a cause or consequence of the disease. And we're not the first one to kind of address this hypothesis. This was put forth, over 20 years ago or more, and books have been written on it. But we have been interested in the role of this for quite some time, and we've tried to model it in animals to see if we can learn a bit more. So if you just focus in on the mitochondria and what happens in mitochondrial dysfunction, we know that a lot of external factors can affect whether mitochondria are healthy and whether they're working properly. And this could include mutations that are caused in mitochondrial DNA, either through a genetic inherited mutation or through mutations that happen by damage to the mitochondrial DNA. You can have glucolipotoxicity, so that's high levels of glucose and lipids. Homeostatic imbalances, toxins can affect mitochondria, many things over time. But we know that the mitochondrial dysfunction that eventually can occur has many different things can happen downstream of that. We know mitochondria are intimately related to apoptosis. So if you have poor functioning mitochondria, cells can be programmed to die. Inefficient electron transport chain activation and activity will inevitably lead to higher production of ROS, which needs to be detoxified. If you have inefficient electron transport chain activity, you have decreased respiration, you have decreased oxidation of fatty acids. So this leads to a buildup of lipid in whatever cell type you're looking at. So these pathways are not specific to hepatocytes. This also happens in muscle and pancreatic beta cell. But many of them have the same kind of downstream problems that can lead in the whole body to effects on blood glucose, free fatty acids, and inappropriate glucose regulation, and then possibly cell death. So when I had first become interested in really the molecular mechanisms that underlie what could be causing this and what are really the downstream effects of this problem, there were a number of studies coming out. Many people were interested in whether mitochondrial dysfunction was inherently preceding diabetes, whether it could actually be a cause. If your mitochondria are unable to adapt to the metabolic stresses that we're putting on our body through the diets we're eating, through our sedentary lifestyle, is it then that we aren't able to deal with this and we eventually develop diabetes? So, Vamsi Mutha and Mary Elizabeth Patty had earlier on, this is an example of some of the earlier microarray studies they had done. And this is in patients, this is muscle from patients with diabetes. So this is diabetes here in this column. You don't need to be able to read this. This is people with a family history of diabetes. So they don't actually have the disease, but it's in their family. And this is people with no family history. And it became apparent that when you started looking at transcriptional gene patterns in these families from these muscle mRNA samples, that the whole program of mitochondrial biogenesis, function, electron transport chain, fatty acid oxidation was downregulated, whether you had the disease or not, actually. So this started to give hints that maybe mitochondria and the inability of mitochondria to adapt to energy demand might be underlying disease pathogenesis, or leading to certain people being more susceptible to the disease developing. And this was interesting to me at the time because I was working on a protein, which is called PGC-1alpha, which is a transcriptional coactivator that regulates the expression of many, many, many of the genes within this list. So this transcriptional coactivator is an inducible, highly regulated coactivator that binds to transcription factors and drives the mitochondrial gene program. It was originally cloned and described 20 years ago by Bruce Spiegelman's lab and others. And it was shown that when you overexpress it in muscle cells, now this is RNA, this is actually a traditional northern blot. And you can see that when you overexpress PGC-1alpha, you can see almost all of the subunits of the electron transport chain will be overexpressed. So you'll have an increase in the entire electron transport chain gene program, which then actually eventually leads to having more mitochondria in cells. These are muscle cells here. You can see more mitochondria, they're denser, they have more developed and dense cristae, and they have a higher metabolic respiratory capacity. So increasing levels of PGC-1alpha will increase the metabolic capacity of cells. A lot of research has been done over the years. This is now quite old, and I modify it as more things come out. But I like this figure because it really illustrates where PGC-1 alpha kind of sits in this process. So PGC-1 alpha sits here. It's a nuclear protein. It binds to a number of different transcription factors. You can see here, the list is probably at about 20 right now, maybe more. And in this process, it binds to these and increases the gene expression of many hundreds of mRNAs that increase mitochondria. It sits in the nucleus, but is regulated by a number of external factors. It's highly post-translationally regulated, and it's also highly unstable, short-lived protein. So it's really an adaptive signal. When the cells need more energy, like in fasting or exercise or energy deprivation, signals are activated, they modify PGC-1 alpha. There's a burst of transcriptional activity to drive the mitochondrial gene expression program and increase the amount of mitochondria in a cell. So I had always had interested in the liver. And while a lot of the original work with PGC-1 alpha had been done in the muscle, there was originally work done in the liver, but what was focused on really in early work was identifying PGC-1 alpha as a regulator of hepatic gluconeogenesis in fasting. So fasting is a signal to the hepatocytes that you need energy. So it's a catabolic signal to break down energy stores to produce glucose for the rest of the body, as well as other forms of energy, such as ketones and fatty acids burning. So what PGC-1 alpha was really known and described for in the hepatocyte was its co-activation of CREB and FOXO1 to lead to gluconeogenesis and glucose production. Work by others and us over the years has then really highlighted the fact that PGC-1 alpha is not just a gluconeogenic signal in the liver, but it's also a signal to burn fatty acids as a source of fuel and to regulate the secretion of triglycerides and lipoproteins into the serum. And this is mainly through its actions on PPAR alpha. So again, it comes down to the fact that PGC-1 alpha is a part of the entire fasting response of the liver. And recently, we've also shown that while it's an important mediator of gluconeogenesis in fasting, it also is an important mediator of the insulin response in the hepatocytes. So its activity primes the liver to respond to that incoming insulin signal on feeding. So what it does is it upregulates the activity by RS2, which when insulin comes in and binds to its receptor and it is activated, it actually very quickly shuts off glucose production. So it's a very tight loop of regulating the fasting response, but also allowing insulin to work better to shut off glucose production in the liver. So it's really a process where PGC-1 alpha tightly regulates glucose production in the liver, either in fasting or feeding, so that glucose levels do not become too high at any point. Why this was interesting to me, of course, I'm interested in metabolic disease, and there were a number of different papers coming out at the time, which consistently showed in humans that PGC-1 alpha levels were low. It didn't matter what tissue you were looking in, whether it was muscle or fat or liver, it was consistently seen that the expression levels of this coactivator seemed to be down. And we thought that in the liver particularly, having low levels of this coactivator might actually contribute to our poor ability to control glucose in metabolic disease and possibly even lead to worsened liver disease. So I became interested really in NAFLD and NASH, so non-alcoholic fatty liver disease. And for those who don't know much about this condition, it affects about 1 in 4 Canadians. It's asymptomatic. Most of us who have it walk around and don't know it. Many people have simple steatosis, which whether that is a bad or good, not a good thing, but whether it is pathogenic, it's not really clear. But 30% of the people with simple steatosis will then go on to progress to develop NASH, which is the more inflammatory state of the liver, state of the disease, which can then go on to lead to bigger problems in the liver. Interestingly, about 50 to 70 or 80% of people that have type 2 diabetes or cardiovascular disease also have NAFLD or NASH. And there's a 2 to 3-fold increase in the risk of developing diabetes if you have NAFLD. So we've really been interested in this theory that NAFLD precedes the diagnosis of diabetes, or precedes the development of diabetes and its role in poor glucose control in this condition. And as I had mentioned, there are many studies coming out now which are really showing that the expression of this coactivator correlates very well with the severity of the disease in the humans. So mechanistically, we wanted to get down to the bottom of whether having low PGC-1 alpha in liver cells could actually potentiate the disease, or whether it was just correlative, whether it was not causative at all. And to address this, we made this mouse model many years ago now, where we lower the level of PGC-1 alpha within liver cells, so within the hepatocyte, using a Krelok system by just 50%. Sometimes we do 100%, but we usually use the 50% model. And this is just to show what it would be like if you only had half of the ability to induce the metabolic adaptation that's regulated by PGC-1 alpha. These mice, when you fast them overnight, you can see that their liver cells accumulate fat or lipid. And this is attributed mostly to their inability to oxidize the fatty acids that are being delivered to the liver from the adipose tissue. If you challenge these mice then with a Western type diet, which is really high in sucrose and fructose and fat, you can see over time, so these are mice that were fed for about 33 weeks on this diet, so it's a really long protocol. And this is oxidative damage to the liver. So this is a 4-H&E stain. And you can see that mice with a 50% reduction of PGC have much more oxidative damage to their liver. When we had a pathologist score the slides, the mice had more fibrosis and had more NASH than their control counterparts. So a 50% loss of PGC-1 alpha led to the development, or potentiated the development, of a worse liver disease in these mice. And when we started to notice at the time, and didn't really understand the link to, was we could also see that the mice with lower PGC-1 alpha on this Western type diet had more inflammation in their livers. From histological analysis of the livers, you could see immune infiltration. And this is not surprising to people, in this field. We know that enhanced inflammation in the liver is usually part and parcel of having fatty liver disease. So we didn't know if this was connected to the fact that these mice had more damage and more fat in their liver and they were just getting inflamed because of that, or whether the higher inflammation we're seeing was actually directly linked to PGC-1 alpha signaling. So we wanted to delve into that a little bit in the lab. And so we researched a bit on the main signals of inflammation in the liver that are linked to metabolic disease. There are many inflammatory signals that the liver sees on a regular basis in life, and there are many chemokines and cytokines. But today I'm going to focus mainly on TNF-alpha and LPS, mainly because these cytokines are are known to be upregulated in the liver in people with metabolic disease and diabetes. And we know the sources of these cytokines can be from either inflamed adipose tissue that accompanies obesity, or increased LPS that can come from the gut microbiome, depending on what type of bugs you have in there or how leaky your bowel is at the time. So the liver is seeing external sources of these cytokines and inflammatory signals, as well as the liver cells themselves are also becoming inflamed. When they have a buildup of lipids, particularly some lipid species are toxic, they will start to produce these cytokines. And one of the common downstream targets of these cytokines signaling pathways in the liver and hepatocytes is the NF-kappa B inflammatory program. So we're not the first to kind of start to get interested in the potential connection between PGC-1 alpha and NF-kappa B signaling. There are many papers out there that have shown this link before and have tried to understand whether or not PGC-1 alpha can regulate directly the inflammatory signaling pathway. Work by Christoph Henschen's lab a number of years back has shown that PGC-1 alpha, if you look at an NF-kappa B reporter system with increasing levels of PGC-1 alpha in muscle cells, you'll see significant downregulation of NF-kappa B reporter. So it seems to be inhibiting NF-kappa B. You can see this at the level of microarray analysis. So in muscle cells that are treated with TNF-alpha, you see major upregulation of NF-kappa B targets in the cells. If you have PGC-1 there at the same time, this signaling pathway is blunted, leading to an overall conclusion, or what I believe is kind of the state of knowledge surrounding PGC-1 inflammation, is that in general, in the systems that have been looked at, it is an anti-inflammatory signal. So increases in PGC-1 will dampen inflammatory signaling, most likely through its inhibition of NF-kappa B through pathways and molecular mechanisms that aren't well described or understood at this point. Also, it can be anti-inflammatory by upregulating some anti-inflammatory proteins that can either feed back on the liver or go into circulation and actually have anti-inflammatory properties elsewhere. What we know is that this is still a signal that's downstream of external signals such as glucagon in the liver or exercise in muscle. And there is some evidence that through things that we don't really understand yet, how TNF-alpha and LPS signaling actually at their receptors on the cell surface also has some crosstalk with PGC-1, although this is not well understood yet. So we became interested in seeing whether the inflammation we are seeing in our mouse models when we reduced PGC-1, whether that was directly downstream of PGC and how that was happening. And we realized through our work with others and through being in the field for quite a while now that we can't think of PGC-1 just as one protein anymore. It's actually at least 9 different proteins. And it actually depends on what tissue you're looking at. If you're looking at in the brain, there's actually 5 more isoforms. So PGC-1 is no longer one protein. So you can't, when you look at it, particularly with overexpression studies, we couldn't just ignore the fact that there were other proteins involved. And in our knockout system, we knock out all of them. So we weren't sure which ones to look at. So what we did first was we went into mouse primary hepatocytes and we decided to say, well, which isoforms are actually expressed in mouse primary hepatocytes. And when we looked at all of the known isoforms and we amplified the whole transcript, we could only find 4. And this is when we stimulate the cells with a stimulus that we know will activate both promoters, which is forskolin. So we could see significant expression of 4 of these isoforms. Many of them are structurally related, like these 2 are very much related. These 2 are very much structurally related, but they're coming from different promoters, as you can see here. We decided to focus our efforts on PGC-1 and PGC-1 Alpha-1 is the one that we know and love, that everybody has worked with for many years. That was the first one that was cloned. Alpha-4 we worked with when I was in Bruce Spiegelman's lab before. We worked on identifying this in muscle with Georges Ruaz, and we've continued our work with it in the liver. So focusing on these particular 2 isoforms, we then went back to our primary mouse hepatocyte system to try to understand whether either of them is also regulating inflammation in the liver. We were able to recapitulate what has been seen in muscle cells before. So this is a NF-kappa B reporter system in primary hepatocytes. This is overexpression of PGC-1 in primary hepatocytes. We add TNF-alpha to initiate the inflammatory response. And you can see that overexpression of PGC-1 will blunt NF-kappa B reporter activity, as was seen in muscle cells, and it will significantly inhibit I think almost all of the pro-inflammatory NF-kappa B targets that we looked at. PGC-1 significantly downregulates them. So this is very similar to what was seen in muscle cells before. When we overexpress PGC-1 to the same mRNA level and add TNF-alpha, we saw absolutely no effect of this on NF-kappa B reporter activity or downstream target gene expression. So it appeared that PGC-1 had no role or no effect on the transcription of these genes or the transcriptional activity of NF-kappa B. But we didn't give up there, because you can only go so far when you're only looking at a certain number of targets. So we decided to do kind of a more unbiased approach by looking at a microarray and looking at overexpressing either PGC-1 or alpha-4 in primary hepatocytes, and we had it either untreated or treated with TNF-alpha for 2 hours. So this is really a short-term treatment with TNF-alpha to look at the initial response. One of the first things we noticed, and it was similar to what we had seen before, is that overexpression of alpha-4 did absolutely nothing in terms of transcription. So really we saw only about 24 genes that were changed when PGC-1 alpha-4 was increased, compared to over 1,000 that we were seeing with PGC-1 alpha-1. However, when you add TNF-alpha, this exploded. So PGC-1 alpha itself, alpha-1, regulated a whole bunch of genes. PGC-1 alpha-4, now all of a sudden we can pick up 3,000 genes that were changing significantly with this protein there, some that were specific only to alpha-4, and many, many that were shared with PGC-1 alpha-1. This is a clustering analysis here of only the PGC-1 alpha targets, and it's really to illustrate, you can see here, if you look at the untreated sample, PGC-1 alpha-4 does almost nothing to the primary hepatocytes itself, but when you add TNF, you can see a dramatic change in the transcriptional program. You can see you can see from the patterns here. So here, PGC-1 alpha-1 and alpha-4 are doing very similar things here as well. And here they're doing similar things, whereas PGC-1 alpha-1 is much better. You can see that alpha-4 is doing a similar pattern. However, there were clusters of genes you can see where it's only a PGC-1 alpha-4 effect that you're seeing. So when we did go biological analysis or go pathway analysis to understand what were these groups of genes. So this analysis was done on these 3 groups right here, either one specific for alpha-1, shared between the 2, or just for alpha-4. And you can see that this group here and this group here in the middle, the ones that are shared and the ones that are unique to alpha-1, really focus around mitochondrial metabolism and catabolic metabolism. So the pathways that these 2 isoforms share focus around the mitochondria and metabolism in the hepatocyte. But the genes that are unique to PGC-1 alpha, we're starting to focus on really pathways that we hadn't seen before that were highlighted from PGC-1 alpha. And so we're starting to see apoptosis, immune responses, and immune signal activation. And these were really specific to alpha-4. We dug a little bit deeper into the analysis, and we looked at this kind of gene set, this small gene set, but 8.5% of the total genes here were regulated by alpha-1 and alpha-4 in the opposite way. So where alpha-1 would increase it, alpha-4 would decrease it. And if you look at just this gene set, what really popped out to us here was the regulation of apoptosis. So where alpha-1 and alpha-4 differed seemed to be in their effects on hepatic cell death and apoptosis. So then we dug deeper into that. So we picked some genes that we had found in the microarray analysis that were related to apoptosis, and that was BRCA2 and TNFa-IP3, which is also known as A20. And these are anti-apoptotic genes that will inhibit cell death when they're overexpressed. And what we saw, we confirmed what we saw in the array, which was that overexpression of alpha-4 in the presence of TNF-alpha will increase the expression of these genes. We also looked at other family members that are related, and we saw similar patterns. Actually, PGC-1 alpha-4 would upregulate this kind of program of anti-apoptosis. And in agreement with what we saw with the microarray, PGC-1 alpha-1 either had no effect or it significantly decreased the expression of these genes. So we were able to confirm that indeed on a certain set of genes from the liver cells, we saw an opposite regulation, and these seem to be related to apoptosis. So in vitro, we then tested that hypothesis. So what we predicted we would see was that alpha-1 and alpha-4 would have the opposite effect on apoptosis. So we did this 2 different ways. One is a Western blot for cleaved caspase 3. The other one is fragmented nucleosomes in the cell. So both of these agree that when you overexpress PGC-1 alpha-1 in the presence of TNF, so everything has to be in the presence of the inflammatory cytokine, we actually saw, if no change, we saw a little bit of an increase in apoptosis when PGC-1 alpha-1 was higher, but overexpression of PGC-1 almost completely blunted the apoptotic effect of this inflammatory cytokine. We always like to complement our overexpression studies with knockdown studies or knockout studies. With the isoforms of PGC-1 that's difficult in many cases because of their homology, mostly in protein structure, in amino acid structures. So alpha-4 is basically a truncated form of alpha-1. They do have different exon 1s, which seem to mediate some of their function. But finding a way to knock down the protein is difficult. We did end up finding a way. So in collaboration with Bruce Spiegelman and George Ruiz, we were able to obtain this mouse and develop this mouse where we knocked out only the alternative promoter of PGC-1 alpha. This of alpha-1. So this would lead to no expression or expression lacking these exon 1s. So you wouldn't get the alternative splice forms that come from this. We tested this to see whether it was actually true. These are primary hepatocytes again. You can see that in response to glucagon, which we know very actively activates the alternative promoter, we know we have very blunted expression of the alternative isoforms. But the ones that are coming from the proximal promoter here, where the expression was fine, if not enhanced, actually. So we think that we had a nice system where we were only knocking out this part of the gene, and we are still having expression from the proximal promoter. So this would include PGC-1 alpha-4, and alpha-1 comes from this side. So we still have alpha-1, we no longer have alpha-4. These are primary hepatocytes. You can see even basally, just in the presence of vehicle, if we knock out the alternative promoter, there was higher levels of caspase-3 in these cells, and in response to TNF-alpha, you see even a more enhanced response than you see in the wild type cells. This was seen at the level of cleaved caspase-3 as well as the fragmentation of nucleosomes. So the loss of proteins from this alternative promoter was leading to enhanced apoptosis in this model. We then went further in vivo. So in vivo, it's difficult to inject TNF-alpha into an animal. It's a very short-lived cytokine. You have to inject a lot of it to induce significant apoptosis in the liver. So another way to do this and to activate similar pathways is to inject lipopolysaccharide, which is something that the liver normally sees in very low amounts from the intestine. But if we inject it into mice, you can induce significant apoptosis into mice. And you can see here that in the wild type mice, we do see enhanced apoptosis. In the mice that are lacking the alternative promoter from PGC-1 alpha, there is much more apoptosis in response to the LPS signal. So which agreed with our in vitro work, and now in vivo, we can also see that the livers of these mice, when they don't have proteins from the alternative promoter, are more susceptible to the inflammatory signals that are circulating in the blood. So like I said, we like to do both gain and loss of function studies. We had done that in primary hepatocytes. We then wanted to do a gain of function study in vivo in mice. There's many ways that we could do this. The way that we chose was we made use of a reagent that we had made in the lab where we can overexpress PGC-1 alpha in any cell type we want as long as there's a Cree mouse available for that cell type. So the way this works is that we knocked in the PGC-1 alpha-4 gene into the ROSA 26 locus, but it's preceded by this stop signal flanked by LOXP sites. So wherever there's Cree in the mouse, this will be removed, the stop signal removed, and you can drive expression of PGC-1 alpha-4. So we did this in the liver of mice. We crossed these mice with the albumin-Cree model as well, the same model we had used for the alternative promoter knockout. Basally, without Cree, you can see that this transgene is a little bit leaky. So we include these controls in all our experiments just to see whether this leaky expression, and we can't control this leaky expression, it's actually everywhere in the body, anywhere that has this transgene, which is every cell. But when we cross it with the albumin-Cree mice, we can see very significant increased expression of the mRNA just in hepatocytes, and you can see this at the protein level as well. So, and the leaky expression wasn't seen at the protein level. So I just showed you this as a representative experiment from these mice, but we were then able, if we just look at the LPS experiment in these mice where we inject the mice with LPS with all the control mice here, we have wild type mice where, you know, if you're not injected with LPS, they have no cleaved caspase 3. LPS will induce significant apoptosis in the liver. You see a similar phenotype in mice that just have the Cree. This is the leaky mouse where we have only a little bit of PGC-1 not at the protein level. And then when you have increased levels of PGC-1 protein in the livers, as you can see, a significant blunting of apoptosis. So we see both in vitro and in vivo, increased levels of PGC-1 seem to inhibit apoptosis. So we think that it's acting as an anti-apoptotic signal in response to inflammation. So another aspect of this whole biology that really intrigued us was this that we had seen before, right? Like, so when we don't have the inflammatory signal, PGC-1 did almost nothing. You know, it wasn't regulating any kind of gene program that could tell us much about what it was doing or to any significant effect. So we wanted to understand why this was. What was it about the TNF-alpha signal that was allowing PGC-1 to gain functional activity? So one way in which this can be regulated is through the localization of the protein in the cell. And for a long time, when I thought of PGC-1 alpha, I thought of it being really as a nuclear protein, and it is. So if you look at PGC-1 alpha 1, which is the canonical form we all look at, and do staining, these are mouse liver cell line or a human liver cell line, you can see that the staining of PGC-1 alpha, which is the green, is exclusively nuclear. Whether you have TNF-alpha there or not, you see it only in the nucleus. And we did cell fractionation as well. And this is actually a good example of a typical PGC-1 alpha antibody that you can buy from many suppliers. I just want to point this out actually, is that many of these bands are very non-specific, and PGC-1 alpha runs just slightly above these non-specific bands. And you can only see it in the nucleus. If you look at PGC-1 alpha 4, it's actually the opposite story, and it explains why it probably has no activity in the basal untreated state. In cells, most of PGC-1 alpha 4 in an untreated cells hangs out in the cytoplasm. So it's more difficult to see in the colored version, but you can see it here, and you can see it in these cells. The majority of the alpha 4 is hanging out in the cytoplasm, but when you treat with PGC-1 alpha or with TNF-alpha, you can see the alpha 4. It's harder to see in these cells because they don't have much cytoplasm, but you can see that some of it moves into the nucleus, and here it's more obvious in the IHH cells, is that you start to see an accumulation of alpha 4 in the nucleus, and you can see this nicely in the cell fractionation. So in cells that don't have any TNF, it's mostly cytoplasmic, none in the nucleus, and then when the TNF-alpha is there, you have accumulation of some of it. And you can still see there's a lot in the cytoplasm, but some of it travels to the nucleus. So we think that what's responsible for this, difference in activation is actually the localization of the protein within the cell. Finally, with this story, we really wanted to understand what PGC-1 alpha was doing once it did get to the nucleus, and was it acting as a transcriptional coactivator like its other related family member, PGC-1 alpha 1. So to do this, we did this in collaboration with George Ruiz at the Karolinska, as well as Jonathan Boulay at the IRCM. They looked at the gene expression array that we had and tried to look for enriched motifs that were in the genes that they had coactivated. We had a number, we had close to 4,000 genes that were changing. So they looked at the promoter regions and the intronic regions and the UTR to see if there were any common regulatory elements. And so if you can see through the color coding where they're looking here. So in the red, this is really the PGC-1 alpha 1 dependent signature. And we were very encouraged to see that we were seeing PPAR gamma, HNF4 alpha, ER, ER1, ER alpha. So these are very traditionally known targets of PGC-1 alpha that we're seeing coactivated in the liver. So this was nice to see. There's also a number of, we only looked at the TNF alpha signature. So in addition to the traditional metabolic targets, we also started to see some inflammatory targets as well. Not as many though, as you can see with alpha-4. So this is the black area here, the ones that are very specific to PGC-1 alpha-4. And what really popped out to us is that we're starting to see stats, STAT1, STAT3, STAT5A. So this kind of group of, transcription factors that are known to be involved in inflammation popped up in this group of genes that were being regulated by PGC-1 alpha-4. If you then look at the yellow group, and particularly the genes that are oppositely regulated by the two different isoforms, again, what came out to be regulated, increased by alpha-4, and oppositely or downregulated by alpha-1 was really genes that were downstream of STAT signaling. And what commonly occurs when we're searching for molecular mechanisms is we then test whether these motifs we saw them enriched. So we took a list of genes that we thought were downstream, some described as STAT targets, and we looked at them to see whether they were differentially regulated by alpha-1 and alpha-4. And particularly what we hypothesized was that PGC-1 alpha-4 in the presence of TNF-alpha would upregulate these or inhibit them in whichever way would lead to an anti-apoptotic function in the cell. But I'm just showing a snippet of what we saw or what we looked at as an example, but we looked at a number of different pathways, a number of different gene targets. And in general, we didn't find PGC-1 alpha-4 had anything to do with the transcriptional level of the mRNAs with or without TNF-alpha. So we weren't seeing what we expected to see. We thought that PGC-1 alpha-4 would act as a traditional coactivator on these genes, but we weren't seeing changes in the gene expression level. So we're still kind of in the dark on how PGC-1 alpha-4 is regulating this gene program. This is an area that we really need a lot more work into at this point to figure out what really is its function in the nucleus. But in summary, we think what our study has been able to illustrate is first, we've been able to describe a new function for PGC-1 alpha-4 in the liver that hadn't been described before there or in any other tissue. And the fact that its localization in the cell could move between the cytoplasm and the nucleus, and that this seemed to regulate its activity. That PGC-1 alpha-1 and alpha-4 have a shared program. And this aspect here was shown previously in muscle. So it is known that alpha-1 and alpha-4 can seem to coordinate some metabolic programs together, and that alpha-4 is generally weaker than alpha-1, but these might be acting together to regulate nutrient and lipid metabolism. But really, we have this separate program for alpha-4, which regulated by inflammatory signals can go in the cell and help to inhibit apoptosis. Whereas alpha-1, in response to inflammation, seems to really do what it's doing in other cells, which is act as an anti-inflammatory signaling factor for NF-kappa B signaling and inhibiting the pro-inflammatory gene program. So of course, we're interested in metabolic disease. So how is this, or is it even involved in the development of NAFLD and NASH? And so we are taking our mouse models, and they have been put on these diet-induced models of NASH, including high-fat, high-fructose models. And we are investigating that now. As you can realize, that these are very long experiments that can take up to 30 weeks on the diet. We really like to go with the diet approach. So these experiments are ongoing, and we're really excited to see whether you knock out alpha-4 or you increase alpha-4, whether it is involved. But what we were able to do in the meantime was to look in human liver samples, and this is in collaboration with Peter Matrakos at the McGill Biobank, where he has biobanked a number of different liver samples from that he's taken during transplant surgeries from rejected livers, and look at liver that's been scored by a pathologist at different stages, either NAFLD, NASH, or liver with cirrhosis, and look at the expression levels of PGC-1 alpha, its isoforms, and the promoters. First, to see whether or not we see even regulation of these proteins in the different metabolic states. You can initially see that if you look at the liver samples that we have from each of the groups, the proximal promoter, so this is the promoter that's responsible for driving the expression of PGC-1 alpha-1 and some of the other isoforms, its expression actually it doesn't really go up. It kind of stays constant, and if not, this is not significant, it's close, but it almost seems to maybe go down as the liver becomes more diseased and fibrotic. That's really speculation at this point. We don't have enough livers to say. But what was clear was that if you look at the transcripts that come from the alternative promoter of PGC, those go up compared to low. So in low, in healthy liver, or in liver that's not steatotic, I'll correct myself because it's not necessarily healthy liver, but you have very low levels of, or almost undetectable levels of these transcripts. But as the liver gets more diseased, you see that some of the transcripts are now expressed in some of the samples, particularly transcripts from containing this exon, and this is the exon of PGC-1a4. And you can see as the liver becomes more inflammatory, this goes up. So it's really the activation of the alternative promoter in human samples that we can see, whereas the proximal promoter doesn't seem to move very much. So we haven't published this data yet, but it kind of leads into some other unpublished data I'm going to show after this to kind of explain what we've been interested in more recently. This is where we amplified the entire full-length transcripts of these to really distinguish between what isoforms we saw in the human liver. And interestingly, the isoforms we see in human liver are exactly the same one we see in mice. So it's just these four. You can see alpha-1 is here, alpha-4 is here, alpha-1 kind of stays constant, and alpha-4 is predominantly expressed. These are different humans. In the more like earlier stages, NAFLD, NASH, we don't see in the cirrhotic forms where we see other isoforms. So while we have those mouse studies continuing where we're looking more at the development of NASH and the inflammatory state of the liver, we've also been doing other studies in the lab. As I mentioned really early on in the talk, we've been kind of interested in this link between metabolic liver disease and cancer. And we know that as liver becomes more inflamed and more NASH-like and cirrhotic, this can increase your chances of getting liver cancer. So 5 to 25% of people with NASH will then progress onto liver cancer. This can actually happen without the cirrhosis step in between in people with metabolic disease. So there is some, link and some need for the inflammatory signaling. So we were wondering how PGC-1 might play a role in this transition here. We've already kind of looked in this transition. We're now looking at this transition. And so again, in these human liver samples that we got from the biobank, we looked at PGC-1 expression. So this is in control livers, from livers that don't have NAFLD NASH. This is from people who have cancer, both of them. This is from the tumor itself, and this is from the adjacent tissue. So in liver tumors of patients, they tended to have lower levels of PGC-1 alpha. If you look at the TCGA database and take the samples from liver tumors, and we separated the patients based on whether they expressed high levels of PGC-1 alpha or low levels of PGC-1 alpha. And this was basically, we took the median of PGC-1 alpha expression, and anything below the median was considered low, and anything above the median was considered high. And if you have low PGC-1 alpha, your survival in HCC was considerably and statistically lower than the people who had higher levels. So it seemed that having low PGC-1 alpha during the development of HCC might not be a good thing. So we wanted to know what that meant actually. So we took our mouse that had either 50% reduction of PGC or a knockout of PGC. We put that on this diet, which is high fat, high fructose for quite a long time. They're obviously not eating hamburgers and drinking Coke, but it's just more to illustrate the diet and its comparison to humans. We also gave them in adult life a small dose of DEN, which is our carcinogen that's biotransformed to the liver, which will predispose them to get cancer. Mice typically don't get cancer of their liver very easily, so you kind of need to give them a little boost. In this model, we can see early on as well as later that exactly what we had seen before in the NASH kind of diet is that mice with 50% reduction of PGC and this protocol have more inflammation in the liver. If you knock out PGC-1 alpha completely, you even have more inflammation in the liver. So there was a dose-dependent increase in the liver inflammation in this model. 4 weeks after we give the DEN, we sacrifice the mice early. This is before they develop tumors. We can see that mice with low PGC-1 alpha or no PGC-1 alpha have higher levels of cell proliferation going on in their livers, which could indicate regeneration or neoplasms forming. But when the pathologist looked at the slide, she identified the fact they did have more neoplasms developing in their liver tissue. And if you leave these mice and let them go for a long time, you'll see that they develop significantly more liver tumors in terms of number and of size. So like I said, we like to do loss and gain of function. At the time, we were still developing the alpha-4 mouse, and we're looking at that now. But we were also thinking about how would we do a more physiological overexpression of PGC-1 alpha protein instead of just doing a traditional kind of transgenic model. And we're starting to think about this polymorphism of PGC-1 alpha that we've been studying a lot in the lab. So it's a single nucleotide polymorphism in PGC-1 alpha that leads to a S to G transition at the amino acid level. About 50% of humans have 2 glycines and 14% have 2 serines. And we'd shown a few years ago that the glycine version of PGC-1 alpha 1, so this is PGC-1 alpha 1, actually, if you're curious, so the canonical form is more stable than the serine form. So the conversion to this amino acid, the glycine amino acid, will stabilize the protein, and the protein has a longer half-life. So in essence, it's like a physiological gain of function model. Mice themselves have a serine at this point. Most animals do actually have a serine. So what we decided to say was, if we change the serine in mice to a glycine, do we stabilize the protein like we see in humans? And in fact, this is true. So this is in vitro. We can see mouse PGC-1 alpha, that's the wild type form, yes. You can see that it's generally pretty unstable. And cyclohexamide, you see it disappear quite fast. Whereas if there's a glycine, if that's the only mutation, you see a much more stable protein. So we made the maps actually using CRISPR-Cas technology. We verified in hepatocytes that changing the serine in mice to a glycine stabilizes the PGC-1 alpha protein. You can see it in a dose-dependent manner here. This is in primary hepatocytes. So when you put this physiologically relevant gain of function model on the same protocol as we did with the knockout, the mice that have 2 versions of the glycine, so that's the more stable version of PGC-1 alpha, have lower tumor number and not significant, but a trend toward the lower tumor size as well. So in essence, in conclusion of what I've shown you today, we've learned really from years of work that PGC-1 alpha not only interacts with glucose production in the hepatocyte, but it really regulates multiple programs that converge on the pathogenesis of fatty liver disease. So while we see roles of PGC-1 alpha in insulin resistance in response to inflammation and dietary sugars, fructose and glucose from the cells, we can see an increase in lipid droplets when you have low PGC-1 alpha due to decreased catabolism of lipids. We have inefficient mitochondria and beta oxidation. We have ROS being produced. Other studies have shown there's ER stress. We've shown oxidative stress. We have higher VLDL secretion from cells that don't have an appropriate metabolic response due to low PGC. And now we have evidence that PGC-1 alpha can also directly regulate apoptosis and cell death in response to inflammatory cytokines. So it's really involved in the milieu of processes within the liver cells that are converging on the pathogenesis of this disease. And now we have new data that's unpublished that eventually having this low level of PGC-1 alpha can lead to worse liver cancer with more tumors that are bigger. So while PGC-1 alpha is really known as a regulator of mitochondrial function, and we originally thought that its role in metabolic disease really kind of focused on this aspect, we now have evidence that it's really involved in multiple aspects of the disease, and that low PGC-1 alpha typically seen in humans actually might be a pathogenic signal that doesn't allow the cells to adapt enough to their metabolic stresses that we put ourselves in. So after all that, I really need to thank the people in my lab who've been involved in these studies, particularly Dr. Melissa Leveillé. What I've shown to you today is really the bulk of her PhD thesis, which she just defended a month ago virtually through Zoom. So I want to congratulate her again on her success in this and really the wonderful story she put together. This has been really work over a long period of time with many people in the lab, past and present. We've had a lot of collaborators and the funding agencies that have really helped us do the work over the years. Thanks so much. I really appreciate everybody tuning in. I'll take questions over Twitter, by email, any venue you want. Again, the Twitter handle is @DNAChicken. You can find me there, or my website, which is diabetesresearch.ca. Have a nice day, everyone, and thanks for tuning in.